So welcome to the second session for Biotechnology for Drug Discovery. In this session, we're going to be looking at producing recombinant products using prokaryotic systems. The learning objectives from this session are first to understand the importance of recombinant protein production in drug discovery and also natural products from microorganisms. We're also going to explore different prokaryotic expression systems, as well as explain examples of products synthesized using prokaryotes and the limitations of choosing the right host and vector system. So why recombinant protein production? Well, we need to overexpress to get enough proteins, and also we need to have easy purification techniques. Now, we can use recombinant protein production in a number of applications, including for functional studies, structural studies. Also, we can use it for developing vaccines, antigens, and antibodies, as well as for development of therapeutic drugs. And also we can use it in industry for industrial enzymes for reactions. So there's a number we're going to focus on prokaryotic cells in biotech production. Now, the origins of producing natural products from microorganisms comes from many efforts by different pharmaceutical companies that have been established over the last century. Sandoz was probably the first pharmaceutical company to receive approval for something called a biosimilar, so this is a drug, in Europe, Japan, and the United States. Ciba, which was a Swiss company that started out in about 1850s as silk dyeing business and branched out in pharmaceuticals in about 1900s. This merged with Geigy AG and then Sandoz and Novartis. Hoechst, which was a company founded in 1863 as the Tefab Ben Fabrik Meister Lewis and Co. In Hoechst, a place called Hoechst, they changed their name and then they merged with Sanofi, Synlabo, and now they became Sanofi Adventis. Bayer, which is probably one of the largest companies in Germany, in the world, it was founded in Barmen in 1863, as well as developing dyestuffs. So they had a factory, and Bayer was probably the first and actually was best known for the product, which was aspirin. Eli Lilly, this is an American company. The company's founder was Colonel Eli Lilly, who was a pharmaceutical chemist and Union Army veteran of the American Civil War. And he started out as a chemist, as a a drugstore in 1869. So in around about 2010, the large proportion of pharmaceutical production actually comes from natural products, enzymes, proteins produced by prokaryotes. The major products that are produced by prokaryotes are antibiotics. And the most important producers for natural products come from different species, including Actinomycetes, myxobacteria, cyanobacteria, and also marine sponges. Whilst biopharmaceuticals are mainly produced in Escherichia coli. The production of natural products by microorganisms. So natural products, there are actually thousands of compounds, and a high proportion come from microorganisms. And Actinomycetes are well known for their high production capacity and are probably most antibiotically active. There are around about 7,000 antibiotics known, with 100 that are produced by microbial fermentation. This picture shows you the applications of enzymes produced from Actinomycetes. And there are different enzymes which are produced by this microorganism. If we start at the top, so I'll just take the laser pointer. There we go, it starts at the top. So here we've got amylases. Amylases are used in fermentation, mainly in the food industry, but also they're used in textile and paper industry. We also have catalase. Catalase is used as an antioxidant. It's also in cold sterilization of beer and also in the dairy industry. Cellulases, these are in kind of biomechanical pulping and also in laundry and also in animal feed industry. We've heard about in the last week L-asparaginase. L-asparaginase has been used in bone marrow treatment, also in stem cell transplant, and also in the treatment of acute leukemia. Lipase, that's being used in something called oleochemical, also in detergent industries, also in diagnostic settings and the pharmaceutical industries. Urase, that's being used in the wine industry, and also the analysis of blood and urine, and also the analysis of heavy metal content in wastewater and soil. Proteases are an important class of proteins. They're used as anti-inflammatory, anti-inflammation. They protect against clots, and also they're in cancer treatment. Chitinase is in plant resistance against fungal pathogen, in drug delivery and wound healing, and also in biochemical industry. So you can see there's an array of different enzymes that can be produced by this microorganism. In addition to these enzymes, Actinomycetes can produce a number of different types of antifungals and antibacterial related bioactive products. Now this table demonstrates some of these bioactive compounds which are produced by Actinomycetes and their related biological activities. So you can see that we've got longifungin, which is this antifungal. So there's a couple of antifungals. We've also got antitrypanosonal. So this is Bacillus. We also have an antiparasitic, ivermectin, antimycin, antifungal, and we also got examples of antibacterial as well. So there's an array of different compounds that this microorganism can produce, which is very useful, obviously, in medicine. So for the production of libraries of natural products, so genetic engineering of microbial strains, so we can do this through introducing deletions into one or several genes, or by heterologous expression of those genes, or combining both of the above. So this might be important for routes for drug design. Now, these technologies have been used successfully to generate libraries of many compounds. So this is kind of like a list of kind of natural compounds that have been used as lead structures for the generation of natural compound libraries. So some examples that you may recognize. So we've got amphotericin, that was produced in S. nodus. We've got a number of antibiotics here, and you can see that and they've been growing over the years. So the production of libraries and natural products. So a recent successful story is about lipopeptides. The story kind of started with cloning and sequencing of kind of clinically useful antibiotics, such as adaptomycin, which is produced by Streptomyces rosesporus. So daptomycin was a novel antibiotic first in the class of drugs. The mechanism of action, so there is a number of steps in its mechanism of action. So daptomycin essentially will bind in the presence of calcium and insert into the kind of the plasma membrane. And then there's like an oligomerization and channel formation, and then you get depolarization and ion efflux. Daptomycin actually causes inhibition of DNA and RNA and protein synthesis and leads to the breakdown of the organism, resulting in cell death. If we look at the kind of pharmacokinetics, daptomycin is actually poorly absorbed orally and actually should be only administered intravenously. Intramuscular injection is prohibited because this causes direct toxicity. And then if we look at the antimicrobial spectrum, daptomycin is a bactericidal antibiotic. It's selectively active against aerobic, facultative, and anaerobic gram-positive bacteria. Also, it's selective against gram-positive aerobes as well. Daptomycin is inherently inactive against the gram-negative bacteria, as they possess an outer membrane that the drug is incapable to penetrate, and they lack kind of specific factors essential for its activity. So other products which include something called Rapamycin, which is produced by something called action planes, Sirolimus, and the cloning of the calcium-dependent antibiotic. Currently, there are around about 50 novel lipopeptides, and Biotech, which is from the Biotech company, formed actually partnerships with leading pharmaceutical companies and developed something called Rapamycin analogues through genetic engineering technologies. Rapamycin can be produced by bacteria, in particular Streptomyces hygroscopus. In fact, it was actually first isolated back in 1972. In fact, the samples were found incredibly on Easter Island, which was quite amazing. Rapamycin is also known as Sirolimus. It's a macrolide compound. It's usually used to coat stents for actually coronary stents. And it's also being used in kind of organ transplant rejection and to treat kind of rare lung diseases. It can inhibit the activation of T cells and B cells by reducing their sensitivity to something called interleukin. Now, rapamycin, the biosynthesis can be accomplished by something called type 1 polyketide synthase. That's in conjunction with a non-ribosomal peptide synthetase. And what you can see here is you've got RAP-A, RAP-B, and RAP-C. These are kind of organized, these are kind of multi-enzymes known as RAP-A, RAP-B, RAP-C. Now they contain modules, and there's up to like 14 modules, as you can see. And these modules, you can modify this kind of chain, develop alternative molecules. So the production of natural products by cloning expression of biosynthetic gene clusters. So biosynthetic genes are clustered in Actinomycetes. The original producers often don't produce more than 1 to 5 mg per litre of compound. So this is kind of very low level of compound. And so you need to express gene clusters in other strains to improve the production rate. So for example, 500 mg could be of something called nicomycin could be produced in Streptomyces lividans TK23, which contain the nicomycin gene cluster. So we can improve the amount of protein or amount of product that we make by changing the organism. Kind of focusing now on prokaryotic expression systems. So an expression system is a kind of genetic construct or a gene encoded by the DNA that are designed to produce a protein or an RNA either inside or outside of the cell. So the expression systems are used in research and also in commercial production of enzymes or therapeutics. And so for overproduction of recombinant proteins, both in eukaryotic and prokaryotic expression systems can be used. So the most widely used system for protein overproduction, both on a kind of laboratory and industrial scale, is the prokaryotic system. Now, this system is based primarily on the bacteria E. coli, although it's increasingly often Bacillus species are used. Kind of looking back through the history, back in 1962, the concept of bacterium was coined by Roger Stainer and C.P. Van Niel. In 1977, Colwers actually proposed to divide prokaryotes into archaea and bacteria. So it wasn't that long ago that we kind of divided the prokaryotes. The lac Operon model that was discovered by some French scientists, Francois Jacob and Jacques Monod, in 1961 from the Pasteur Institute. And they actually were the first to show the organization of bacterial genes into operons through their studies on the lac Operon of E. coli. And they won the Nobel Prize in 1965, it's a Nobel Prize for Physiology. So in 1977, Herbert Boyer and Arthur Riggs described the first vector, and the first vector was called P, which P always stands for plasmid. BR is normally, sometimes it's the people who invented it, so they took the second name, so Boyer and Riggs, so B was for Boyer, R was for Riggs, 322 plasmid, which is widely used as an E. coli cloning vector. So when we kind of look at different kind of prokaryotic gene regulations, there are different types and components of prokaryotic gene regulation. So we kind of discussed kind of briefly about operons. So operons are clusters of genes that share the same promoter and are transcribed as a single large messenger RNA that contains multiple structural genes or cistrons. The trip operon, which is a repressor operon, that's found in E. coli bacteria, is a group of genes that encode biosynthetic enzymes for the amino acid tryptophan. And actually the trip operon can be turned on or turned off. So when it's turned on, that's when the tryptophan levels are really low, and it's turned off when the tryptophan levels are really high. Another regulator is the catabolite activator protein cap. It's an activator regulator. It's also known as CAMP receptor protein, and it's a transacting transcriptional activator that kind of exists as a kind of homodimer. Continuing on about prokaryotic gene regulation, so the DNA prokaryotes is organized into the circular chromosome, which is kind of supercoiled in the nucleoid region of the bacteria's cell cytoplasm. You also have free DNA as well. The proteins that are needed for specific function are encoded together in blocks called operons. And for example, all the genes needed to use lactose as an energy source are encoded next to each other in the lactose or the lac operon. So we're kind of focusing on the prokaryotic gene regulation. So in prokaryotes, there are structural genes related to function that are often organized together on the genome and then transcribed under the control of a single promoter. So you can here you can see we've got the regulatory gene. We have a single promoter, and the operon regulatory region includes both the promoter and also the operator, so within the operon region. So if a repressor binds to the operator, okay, so if it binds to the operator, then the structural genes will not be transcribed. Alternatively, the activators may bind to regulatory regions. This enhances transcription. Okay, so if you get binding to the operator, then you'll get a block of transcription, but here you get an enhancement. And these are normally before the kind of structural genes. Okay. So there are two different types of gene regulation. So you can get positive or negative. Okay. And the activators, or sometimes the inducers, instigate kind of positive regulation, whilst the repressors instigate the negative regulation. And when an activator or inducer binds to the operon, the transcription process either increases in rate or is allowed to continue. When the repressor binds to the operon, then the transcription process is slowed or halted. It's kind of like a visual now, just to have a look at this. So the types of regulatory molecules. So we have three types of the regulatory molecules that affect the expression of the operons. So we have these, the repressors. These are proteins that suppress the transcription of a gene in response to an external stimuli. So in other words, the repressor keeps a gene turned off. Whilst the activators are the proteins that will increase the transcription of a gene in response to an external stimulus. In other words, the activator turns a gene on. And then we've got the inducers. Okay, so these are small molecules that either activate or repress transcription, depending on the need of a cell and the availability of a substrate. And inducers basically help to speed up or slow down on or off by binding to the repressor or activator. In other words, they don't work alone. The cistron. So the cistron is a genetic unit coding for a structure of a subunit of a protein molecule. A single messenger RNA carries information for multiple proteins. Okay, and the type of messenger RNA is called a polycistronic messenger RNA, and is totally unique to prokaryotes. So prokaryotes have a polycistronic messenger RNA, which can actually produce multiple proteins, whilst eukaryotes have a single, so it's a monocistronic messenger RNA, which then produces just a single protein. The polycistronic lac operon messenger RNA is then obviously translated into these kind of three different proteins. So the lac operon, this is an inducer operon. So the lac operon, which is the lactose operon, is an operon required for transport and metabolism of lactose in E. coli and many other enteric bacteria. Although glucose is actually the preferred carbon source for most bacteria, The lac operon allows for effective digestion of lactose when glucose is not available through the activity of beta-galactosidase. Now, the type of gene regulation in prokaryotic cells occurs through inducible operons, and the lac operon is a typical inducible operon. Now, the lac operon encodes genes necessary to acquire and process the lactose from the local environment. Now, there are a number of components of the lac operon. There are a number of genes, so we've got here LAC1, Z, Y, A, and these, and then also we've got promoter and operator. Now, they all have different functions. So LAC1 is a constitutive gene which synthesizes lac repressor constantly, whilst LAC-Z is a gene for beta-galactosidase subunit. LAC-Y is a gene for the permease subunit. LAC-A is a gene for the thiogalacosidase transacetylase subunit. The promoter, that's the site for RNA polymerase binding and is the initiator of transcription, whilst the operator is the repressor binding site. So if you have molecules which bind to the operator, this will repress the transcription. So let's moving on to prokaryotic expression systems. So we're going to focus first on E. coli. E. coli, it's a bacterial host. It's probably the most important and widely used in the industrial pharmaceutical protein production. Now, there are a number of advantages of E. coli. Firstly, it's really easy to handle. The media it's not very expensive, so we can culture bacteria quite easily. We know about the genetics of E. coli. We have a number of genetic tools available which we can genetically modify the E. coli. It has a fast growth. It can divide every 20 minutes. And also there's a potential to have a high yield of recombinant proteins, normally between 20 and 30% of total cellular proteins. There are a number of several disadvantages of using E. coli, and we'll come to this a little bit more when we discover about the eukaryotic systems. One of the first ones is something called acetate formation in high density cultures, which can lead to cell toxicity. There's also a lack of post-translational glycosylation, which sometimes is important for certain proteins which need to be glycosylated, and so E. coli don't have the facility to glycosylate proteins. Also, you can have an accumulation of endotoxins. Proteins can also form inclusion bodies, and they may require refolding, which E. coli may not have the ability to do that. But there are now adaptive strains, and we'll discover about those adaptive strains. Also, proteins with disulfide bonds may be difficult to express in E. coli. There's been a number of advances in E. coli production systems. Now we have different promoters to regulate the expression. We can control oxygen levels. We can optimize specific growth rates. We can even co-express certain chaperones. We can even secrete proteins into the periplasmic space or into the media, which is often favourable for protein production. We can get different mutant E. coli strains, which have been developed, which show specific features to meet special demands of particular proteins. Focusing now on the first E. coli strain, which is known as E. coli BL21. This is like a schematic view of the 4 types of E. coli strains. So we've got BL21 star, Rosetta strain. We've got Shuffle and Origami. These are used in recombinant protein production. Now, BL21 are from E. coli B strains, but they have defects in genes known as the LOM protease and also OMP-T protease. This picture shows us that these strains actually can have some leakage, so we can have a kind of leaky expression. So when we express, so if we transform our vector, so this would be a vector such as like the PET vector. What happens is we can actually have leaky expression of the T7 RNA polymerase. And one way to address this problem is to use 1% glucose added to the media. So by adding the glucose to the media, this may be good because the proteins may become toxic to the bacteria. What ways that we can induce the expression of our particular genes is to use something called IPTG, which we'll come to in a moment. That's only used to express proteins with a kind of T7 promoter in the upstream of their genes. This table actually shows some of the common E. coli expression systems. So we talked about BL21. BL21 has a number of features. It has something called the DE3 lysogen that expresses this T7 RNA polymerase. It is also deficient, as we said, in ion and OMP-T proteases, and also can be induced with IPTG. So IPTG is used for inducing expression. It's suitable for the expression of non-toxic genes and requires growth conditions of 1% glucose in the media. So you can see here, these are the companies that produce these different strains. So Novogen is quite a famous company that has developed a number of E. coli bacterial strains for protein expression. An adapted strain is known as BL21DE3 P-lyse. This has the DE3 lysogen that expresses the T7 polymerase, but it also has a T7 lysozyme to decompose the enzyme of the T7 polymerases before induction. The benefit of this strain is that it prevents leaky expression and is suitable for the expression of toxic genes. The growth condition of this particular strain is in the presence of chloramphenicol at 34 micrograms. Now there are a number of strains, so we've got BL21, so you have mutation in a gene called RENE131. There's messenger RNA has more RNA stability. Oregano strains, these are particularly great. It has a mutation in something called the TRXB and GOR genes. This actually enhances disulfide bond formation in the cytoplasm. So this is actually quite a nice strain if we want to fold our proteins. So you can see there's different strains. Don't worry about kind of concentrating on those strains. We use the different strains for different applications. So this is really like a summary of a number of E. coli strains and their applications. The most common strains that we use for kind of routine cloning or subcloning and also blue-white screening are XL1 or DH5 alpha or top 10 E. coli. So these are the popular ones that you might have come across in the past. Now there's some other strains which are very useful for kind of high efficiency cloning. So for example, for library construction, XL10 gold and MegaX and DH10B are useful for this. For cloning of unmethylated DNA, so if you don't want your DNA to be methylated, we can use the XL1 blue MR strain. Again, for production of unmethylated DNA, we can use the JM110 or IMV110. These strains here for cloning unstable plasmids, so SURE and STBL4. The expression of T7 promoter, so this is for the production of proteins. We have the BL21 DE3 or the BL21D3P lyse, that's for the expression of T7 promoter to reduce kind of leaky expression, so we have a tight regulation of expression. The other strain that we've kind of looked at is BL21 codon plus rosetta. This is for the expression from a T7 promoter with a codon biased correction. And the Origami strain, this is useful for improved disulfide bond formation. And MAC1 is useful for fast cloning due to quick cell growth. Other types of bacteria that we can use for protein production, it's more common to use E. coli, but we have seen in the past that some companies are using now Bacillus. Bacillus is good for expression, particularly of enzymes such as proteases for detergents, and also amylases for starch and baking. Bacillus is not currently used for therapeutics, so mainly E. coli is used for therapeutics. The advantages of Bacillus is that it has a high capacity for secretion of proteins. There's no lipopolysaccharide, LPS, containing outer membrane. There's a metabolic robustness. There's also it's genetically well characterized, and you can also get good transformation and gene manipulation technologies. Now, the common species of Bacillus are B. megatherium, B. subtilis, B. licheniformis, and B. brevis. Now, Bacillus bacteria is a gram-positive rod-shaped bacteria. Moving on now to expression vectors. So there are many different commercial expression vectors available. And for successful production of foreign proteins in E. coli, this depends on 1, the host strain that we're using, 2, the fermentation conditions, and 3, the expression vector. So it's an important aspect to consider when designing our protein production experiments. Now, the vectors for protein expression, they have a number of features which we'll kind of visit. In particular, they have something called a selectable marker, so this is allowing to select for the vector. We have also regulatory genes, which allow us to regulate the expression, so known as the repressor. We have also origin of replication, so remember bacteria tend to have 1 origin of replication, whilst humans have thousands. There's always a promoter, so the promoter strength may want to have it to be weak, and so it's kind of slowly produced, or you may want to have a high strong promoter where it's kind of quickly made. There's also a transcription terminator, something called a Schrein-Dalgarno sequence. We also got start codon, tags and fusion proteins, also protease cleavage site, and multiple cloning. So you'll come across these terms a lot when we talk about vectors. Now, these vectors, the PET vectors, are probably the most famous vectors of all. A company called Novogen, they actually introduced PET vectors, and actually this is probably the first choice when we come to doing cloning and protein expression projects. The vector tends to have a very high expression, and that's due to a potent promoter. And it's able to, so you've got this promoter which is like the T7 promoter. You can also add tags, so things like his tag. So you can see here you've got his tag in this PEP16 vector. Here you've got 2 other vectors called PET39 and PET40. They're slightly different. This one's got different restriction sites compared to the PET40. Now you can see there's a his tag, and that's really useful when we want to come purify the protein. And the his tag can be put at the C terminus or at the N terminus of the protein. And the proteins can be overexpressed even without the tag as well. The his tag may alter some enzymatic properties of our target protein, and that may be something that we may need to consider. Whilst the his tag may actually reduce the solubility, which may be a useful feature. Looking at other types of PET expression systems, so this is an example I just pulled off the internet. So this is from a company called Prokaryotes. They've made this, it's called a PET Express and Purify kit. It's an infusion ready, so I think it's kind of fast cloning. The PET Express kit allows you to express a protein of interest with an N-terminal or C-terminal his tag in E. coli. And what you can do is you can clone with a T4 DNA ligase. So you clone with infusion, so you clone your gene of interest into the PET vector. Afterwards, then you transform the construct into E. coli to express your protein, and then after we can then purify, so we grow up the bacteria in kind of like a small culture, and then we expand down into a larger culture, and then we can purify using a special adapted column. So here they've got a his talum purified protein column, or you've got the nickel purified column. Something to bear in mind is the replication rate. So the origin of replication is located on the plasmid, and it determines the plasmid copy number in the E. coli cell. Now, a plasmid copy number essentially gives us an idea of gene dosage, and also this is accessible for expression. So one would say that with a higher plasmid copy, we get higher yield of protein, whilst low copy numbers of plasmids lead to lower translation rates, which may be useful for protein folding. Now, a little bit about kind of the chaperone systems. These are kind of like best characterized chaperone systems in the cytoplasm of E. coli. It's known as the DNK, DNJ, GRPE, and GROEL and GROES. And so to support the folding process of certain plasmids, we can actually co-express these chaperones in which genes of bacterial chaperones are encoded. We can express these, and this may actually improve the folding up of our protein. When we don't have proper folding, we may get like misfolding. And so if we do get like partially folded or misfolded protein, we may be able to refold the protein using the DNJ, GROEL and GROES system. Here we were looking at kind of promoters. There are different types of promoters depending on strength. We can have strong promoters, or we can have weak promoters. So these are examples of E. coli promoter systems used for heterologous protein production. They may also be inducible. So it may be that they're inducible or constitutive. Okay, so we talked about IPTG. So IPTG stands for isopropyl beta D1 thiogalactoparacid. It's a chemical reaction which kind of mimics something called allolactose, which kind of removes the repressor from the lac operon. So remember, when we have the if we have the repressor on the lac operon, this it removes it. It helps to induce gene expression. And there are different kind of lac promoters. So, for example, the lac promoter is IPTG, is inducible. We've got the lac UV5 promoter, T7 RNA polymerase promoter lac. We got the TRC and TAC promoter, and also the ARA-BAD promoter, and these are all IPTG inductible. We've also got the TET-A promoter. This is, I'm sorry, I made a mistake there. ARA-BAD promoter, this is L-arabinose inducible. The TET-A promoter, that's tetracycline inducible. This actually has been, the tetracycline has been used in mammalian cells. The TRP promoter, that's tryptophan starvation, is induced by tryptophan starvation, and the Pho-A promoter is induced by phosphate starvation. So IPTG induction. So the lac repressor protein that's evolved to sense for the presence of lactose, and that's combined with galactose and glucose disaccharide. Both the host chromosome and the insert have copies of the lac repressor and the lac repressor gene, so that's to ensure that there's always enough lac protein available. In the absence of lactose, the lac repressor binds to the operator sequence on the DNA, and what it does, it kind of bends the DNA by like 40 degrees. And this kind of blocks the access of the T7 RNA polymerase to the promoter site, and this might prevent the leaky transcription of the gene before induction. When the lactose binds to the lac, it induces a conformational change in the protein structure. That kind of renders it incapable of binding to the operator DNA sequence. Now, IPTG, that mimics the structural. It mimics the structure. It's a kind of structural mimic of lactose. Okay, and it also kind of resembles galactose sugar, and it binds to the lac repressor and induces a similar conformation chain. So when we induce our cultures with IPTG, what we observe when we run our samples or our our cell lysates on a gel, what we observe is, like in the non-induced sample, we see kind of a number of bands here, but there are no kind of very significant bands. When we induce with IPTG, we can induce the expression of our protein. And here you can see the example here of a protein which has been induced. It's about 20 kilodaltons in size. Moving on now to tag fusions. So tag fusions, for example, we've got maltose binding protein, MBP, thyrodoxin, TRX, glutathione S-transferase, GST, polyhistidine, and also we have things like GFP tags, flag tags, all sorts of tags. The benefits of using tags is that you increase the protein stability and solubility, but also you facilitate the purification. In particular, the maltose binding protein, thyrodoxin, and GST, they improve the stability and solubility and purification of our proteins. Whilst the polyhistidine GFP tags, they're important for kind of purification and detection. And there are a number of disadvantages. However, we may need to use proteases to free the protein, and this can be quite expensive. And also cleavage may be rarely complete, and hence there's an effect upon the yield. You can have a look at some of the advantages and disadvantages by clicking on this website. So using tags to isolate proteins. So some expression vectors have DNA sequences that kind of encode these short protein tags, and the his tag is probably the most famous of those. Now the his tag is normally found as 6 his tag codes, which code for 6 histidine residues, and they're fused in frame within the coding sequence for a clone gene. And the tag is then fused to the protein. So the tag is made along with the protein. So essentially what you have to do is you have to remove the stop codon. So you remove the stop codon and allow for the translation of the tag. So the tag is then added either to the N-terminus or to the C-terminus of our protein. So by adding the his tag to the N-terminus or the C-terminus, we can then use this tag for the purification. For the purification, in particular for the his tag, we can use a nickel column to bind the His tag, and we can then purify the His tagged proteins. Now there are other types of tags that we can use to isolate our proteins. These are more kind of focused on epitope tags. In particular, we've got the MIC and flag tag. These are kind of specific antibody epitopes that kind of work in a similar way to the His tag. Now the MIC tag and the flag tag can be isolated by binding to the antibodies, such as the MIC or the flag, but they work in a similar way to the His tag. So focusing now on the selectable markers. So in the absence of selective pressure, plasmids can be lost from the host. So we need to select for our plasmids. And this is actually really true for high copy number plasmids, or when the plasmid-borne genes are kind of toxic to the host, they'll be kind of got rid of. And the way to do this is to kind of use these kind of selectable markers. Now, you've probably come across in the past the famous one called ampicillin. Ampicillin is an antibiotic, and when ampicillin is present, and if the vectors doesn't have ampicillin, then you will kill the bacteria, and normally that's used at a concentration of about 100 mg per ml. There's another type of, it's a bit like ampicillin, it's a bit kinder, it has a similar structure to ampicillin, is carbonicillin. Again, that's also used at 100 mg per ml. Other types of antibiotics are chloramphenicol. That's kind of a special antibiotic. We normally add this, this actually will be coated on the plates as opposed to added to hot media. That's used at 34 mg per ml. We also have Actinomycetes, Rapamycin, and tetracycline. So there are some other features that we kind of we've mentioned before, but we'll discuss further now. And there's one which is called the Schrein-Dalgaro sequence. Now the Schrein-Dalgaro sequence is required for a translation initiation, and is kind of complementary to the 3' end of this 16S ribosomal RNA. Now, the efficiency of translation initiation at the start codon depends on the actual sequence. So it depends on a consensus sequence of something called has a 5' sequence of T-A-A, G-G-A, and G-G. And so that's kind of a very important sequence. Now we also have start codons and stop codons. So the start codon is obviously the initiation point of translation, and in most E. coli they use the ATG, which is normally codes for methionine. However, there are other start codons in E. coli which are not hardly used, so such as GTG, which is around about 8%, and also we've got TGG and TAA. The stop codons, these are kind of termination of translation. There are 3 possible stop codons. So we've got TAA, it's a preferred one because it's kind of less prone for read through than the TAG and also TGA. We've also got the multiple cloning site. So the multiple cloning site is essentially a series of unique restriction sites that enable you to clone your gene of interest into the vector. And you also have something called a protease cleavage site. So this may be useful to remove the tags. So the protease cleavage site is often added to be able to remove the tag or the fusion protein from that fusion protein after expression. Some of the common proteases are listed in this table. So we've got factor XA, enterokinase, thrombin, TEV protease, and precision. Kind of focusing now on messenger RNA instability. This can be an issue with E. coli cultures. Environmental changes can cause messenger RNA degradation rates to change kind of as a global or gene-specific way. And bacteria kind of adapt to many stresses and also changes in the environment. So that kind of involves modulating the degradation rates of specific transcripts, which encode obviously the proteins which are relevant to changing the conditions. So some stresses, particularly those which cause like severe energy stress or trigger kind of global stabilization. So we get different types of stresses. They may change or cause the degradation, and so you get like instability of the messenger RNA. And that might be related to kind of fast, say rapid growing bacteria or slowly growing bacteria. This may actually alter the protein production, so something to bear in mind. Also, we've got kind of small RNAs, which can affect the messenger RNA stability, and this is through it's been proposed through different mechanisms. Here, the small RNA binding can actually mask preferred RNase cleavage sites, so therefore kind of stabilizing the transcript. Okay. In this case here, we may get the small RNA may bind, which can block ribosome access to the shrine Dalgarno sites, so reducing the translation and also typically the destabilization of those transcripts. And in C here, the E. coli and maybe some other gram-negative bacteria, the pairing of something called the small RNA and the messenger RNA can be mediated by an RNA chaperone known as HFQ, which kind of leads to the RNA degradation. We're going to focus on some other types of cloning. So there's one which is called Recombinering, which can increase the speed of cloning. Essentially, you can assemble new DNA vectors with different genes of interest that can be difficult when the gene is long. So this can be quite hard to identify unique restriction enzymes which are compatible with the polylinker. Large recombinant DNA vectors can be created using homologous recombination. It's a process called Recombineering. So essentially what we can do is we can take our gene of interest and we can electroporate essentially our gene of interest in this kind of linear template. Here we have 30 to 50 sequence homologous to the back here with the gene, and this is kind of flanked either side. This can be electroporated into, so the gene sequence can be electroporated into bacteria. What happens is that the bacteria can actually you can get homologous sequence to the ends of the gene of interest, so you can get kind of recombination of the gene for the lambda red, which is called recombinase. This is temperature sensitive, so the gene of interest can be flanked, and once it's inside these red proteins, they kind of recognize the ends of the gene of interest, and they facilitate the homologous recombination. So that's between the back and the gene of interest, and then the red proteins then are produced only when the bacteria are exposed, and this is normally triggered by high temperatures. And when you drop the temperature, say to 32 degrees, this will stop the red proteins from working, and so here you would have then the recombined vector. So that's kind of a very quick way of generating your vector, and this may be useful for when you have very large genes that you need to clone. For the selection and counter-selection in this kind of recombinering system, recombinant vectors can use a selection counter-selection method to essentially to identify which bacteria harbour the vector containing the gene of interest. The GALK, which that encodes something called a galactose kinase, when the bacteria are grown on 2-deoxygalactose, the toxin is produced and killing the bacteria, whilst the bacteria can be grown on galactose minimal media. Okay, so when you have the vector without the gene of interest, when we clone the gene of interest through the recombination, so Recombineering replaces the GALK gene with the gene of interest, the bacteria cannot grow now on the galactose minimal media, whilst they can actually now grow on the 2-deoxygalactose, and that's because you've got rid of this GALK gene. We also have something called gateway cloning vectors. Now, gateway cloning technology has been based on site-specific recombination system, which is kind of used by phage, and that's to integrate the DNA into the E. coli chromosome. Both the organisms, so the phage and the bacteria, have specific recombination sites known as ATTP in the phage site 1, and also ATTB in the bacteria. There's like a integration process known as lysogeny, which is catalyzed by two enzymes. There's a phage 1 which is encoded protein known as integrase, and the E. coli protein is known as IHF, which is an integration host factor. Upon integration, then essentially these sites here will flank the prophage gene insertion. Now, the gateway vectors have a number of benefits. So they have the origin of replication. There's also selection through Rapamycin. We also have multiple cloning site known as the CCDB site. So the CCBD, that actually encodes a toxin that can kill the host bacteria. Okay, and if we clone within this site, the CCB, between the two, so ATTL sites, the gene of interest then replaces this CCDB gene. Moving on now to production steps. So the production of biopharmaceutical drugs is kind of strictly regulated and is based on the directions given in the European Pharmacopoeia. Generally, the following steps are involved. So 1, we have the development of a host vector system and its detailed characterization. We also have the production of a validated master cell bank and derived working cell banks. We have fermentation, then protein isolation after fermentation, and then once we've isolated our protein, then we need to purify the protein. There's also then analytic testing done extensively, and also evaluation of the protein product to check that it is in its correct structure. And then you have a detailed documentation of each single step. Now, there are around about 140 biopharmaceutical products that have been approved by EMA, which you can actually find updates on these websites. 27 of these are actually produced in E. coli. The following table shows you a list of some of those recombinant proteins which have been used in human therapeutics. In particular, we've got things like cytokines, antibodies, we've also got insulin, we've also got interferon. What else do we have here? We've also got something called keratinocyte growth factor. We've also got hormones, so parathyroid hormone, cytokines. We've even got vaccines which have been produced, stomatotrophon, etc. Okay, so actually looking at stomatotrophon, I've just mentioned the last example, somatotrophin known as STH or human growth hormone or HGH. So human somatotrophin, this is a peptide hormone of 20 kilodaltons, and the active hormone is around about 191 amino acids, has 2 disulfide bonds, and is not glycosylated. Now, it's normally synthesized in the anterior pituitary cells as a prohormone. So it has this amino terminal signal sequence, which is normally cleaved in the endoplasmic reticulum. The E. coli is used as a host system. So how do we produce somatotrophin? Well, how do we produce any gene or any protein using E. coli? So with somatotrophin, we can take our gene for the human growth hormone. First, we need to extract RNA from the human cell, and then we perform something called cDNA synthesis and then reverse transcription. Afterwards, we can then clone our gene for the human growth hormone, and when we perform a PCR, we use E.coli to incorporate restriction sites at the 5' and 3' end of our gene. Then we can cut with E.coli to create these sticky ends, and then if we take our vector, so this will be, say for example, the pet vector that we've seen before, we cut with E.coli and then we can add in our gene to generate the gene for human growth hormone. And then what we can do then is transform the bacteria, and the bacteria itself will contain the gene for human growth hormone. And then we can induce the expression using, for example, the IPTG induction. So somatotrophin, there are different kind of strategies used to express and isolate the hormone. So that was just really simple strategy. But other companies like Pharmacia and Pfizer have made something called ginotrophin, somatotrophin. That's expressed as a fusion protein, which contains a signal peptide to direct the protein to the periplasmatic space. Now, the host strain is derived from the E. coli K12 and expression vector PBR322. Now, the PBR322 is composed of the following elements. So you've got col E1 replication origin. You've also got the tetracycline resistance genes. We've also got the beta-lactamase gene, and also fusion promoter, which is made up of different parts of the promoter, the TRP promoter, and also part of the E. coli enterotoxin 2 signal sequence, so allow it to be targeted to the periplasmic space. And also you've got the complete cDNA of the human somatotrophin. So combining the two parts of the promoter gives us a kind of efficient transcription of the target gene. The enterotoxin signal sequence helps to direct the fusion protein to the periplasmatic space by peptidase. The peptidase removes the signal sequence. The host system is transformed by using calcium chloride competent cells, and that's based on a single positive colony. So we select a single positive colony, which then we use as our master working bag. And then we can aliquot this is then used to inoculate a preculture. And the preculture is then incubated on a shaker and expanded as a seed culture. And the seed culture is then used to inoculate then a production fermenter. And at the end, the cells are then harvested by centrifugation, treated by freezing at -20, and then thawing. And then the cell wall ruptures, releasing the periplasma containing accumulated somatotrophin. And this somatotrophin then is purified using several chromatographic steps. And here we've got the final product of genotrophin. In our second example is human insulin. Now, human insulin, that's normally that's a mature insulin. It's composed of two different peptide chains. It's normally translocated to the ER, and in the ER there is a removal of the signal sequence, so kind of giving rise to something called pro-insulin. Now, the cleavage of the internal 32 amino acids fragment generates something called the C peptide, and also the A chain, which is 21 amino acids, and the B chain, which is 30 amino acids. Now, the two chains are held together by two disulfide bonds. Here you can see this is the insulin, which is the A chain, and the B chain, which are held together by these two. You can see in the yellow here, these are the disulfide bonds between those two chains. Now, the third disulfide bond is located in the A chain. The beta cells are the pancreatic isolates. Insulin is normally synthesized as a single chain of 86 amino acids, precursor polypeptide called pre-pro-insulin. The production of insulin is challenging, and that's due to its structure. So E. coli are not able to perform the post-translational modifications. Insulin chains were made separately, so you can make them separately in different E. coli strains and then synthesize as a fusion protein using beta-galactosidase. The chains are then liberated and then purified and then separated from the beta-galactosidase and then joined by chemical and chromatographic methods. The efficiency and yield was very low. However, today, now we can take the insulin gene without the signal sequence. It's expressed under the control of the TRP promoter. You get like a chimera of the amino acid terminal end of the tryptophan synthase, which is joined to the proinsulin atomethionine residue. So we can insert the genes for each of the two insulin peptides next to the highly expressed gene. And then we perform a transformation, transform the E. coli with the recombinant expression vectors and select for the transgenic cells by antibiotic resistance. Afterwards, we then purify the fusion protein. So here we've got the A chain and the B chain, and they're purified separately. Then we can chemically remove the insulin polypeptides from the beta-galactosidase protein and then recombine, kind of facilitating the kind of disulfide bond formation, combine the polypeptides to produce a functional insulin. So in summary to the session today, we've looked at there is a large proportion of pharmaceutical production which comes from natural products and enzymes and proteins produced by prokaryotes. Expression systems are genetic constructs, a gene encoded by DNA that are designed to produce a protein, or an RNA, a ribonucleic acid, either inside or outside of a cell. Expression systems are used in research and in the commercial production of enzymes and therapeutics. Now, the most widely used system for protein production, both on the laboratory and industrial scale, is the prokaryotic system, and this system is primarily based on the bacteria E. coli, although increasingly often Bacillus species are used. Some further reading to take a look at, in particular this paper from Hayat in 2018, is looking at recombinant protein expression in E. coli. What we need to know, it's quite a nice paper. Also the chapter from Kaiser Pharmaceutical Biotechnology, Drug Discovery and Clinical Practice, so chapter 2, have a look at this between pages 15 to 25. And another paper looking at recombinant DNA technology and DNA sequencing. This is available from the authors of Mark and Roberts in 2019.